एक बार पूछ लीजिएगा कि एक बार पूछ लीजिएगा।
Good morning. Namaskar. Welcome to Hortalk-54. Today's speaker is Dr. Nagendra Rai. He is from Principal Scientist ICAR-IIVR Varanasi, UP. He will speak on traditional and molecular breeding approaches to manage abiotic stresses in tomato. On behalf of Progressive Horticulture, I, Shiv Pratap, welcome the speaker Dr. Nagendra Rai and you all to this edition of Hot Talk. Now I request Dr. Nagendra Rai to start the talk. Sir, please.
So, thank you very much for giving me an opportunity. I am also thankful to the organizer of the Progressive Horticulture. They have given me opportunity to express my 30-year experiences on tomato. And the topic which I am giving to present here is also a part of my research of the 30 years. The topic which I have chosen, that is a traditional and molecular breeding approaches to manage the abiotic stresses in vegetable crop. In tomato, I regard the tomato, which is considered as a ideal research material, vehicle materials or materials, because it is easy to culture, has a short life cycle, high cell fertility and homozygosity, great reproductive potential, high amount of उन्हीं का
सर कैरी ऑन कीजिए सर.
अच्छा कैरी ऑन करें.
यस सर. यस सर.
स्टार्टिंग से कि जस्ट स्टार्ट से कि 20 से. हेलो
आप. यस सर.
should I start from the beginning or the portion which I have left?
No, you left that portion.
अच्छा. So that we have discussed about the tomato have a high cell fertility, homozygosity, great reproductive potential, high amount of the genetic diversity are also existing. In case of tomato, more than 1,300 genes have been described, and out of which 242 have been assigned to a specific location. They all are situated on the tomato chromosome, because in case of tomato, the N chromosome is 12. Normally, tomato is a self-faulted crop, but natural cross-pollination also occurs. In case of the California, they have reported that it is a 1.9 to 47.1% natural cross-pollination occurs, whereas the southern Bureau, they have reported that the natural pollination occurs 15 to 20%. Globally, tomato is being grown in 3.7 million hectares with the production of 200 million tons. Mexico, Brazil, Italy, and Spain are the top producing countries in the world. India is the second largest producer in the world, which contribute 9% of the major vegetable crops. In India, 13 states are the major producing states, out of which Andhra Pradesh, they contributed 13.99%, whereas in Madhya Pradesh, it contributed 12.24%, and Karnataka contributed 10.99%.
कुछ टेक्निकल की वजह से स्लाइड शेयर नहीं हो पा रहा है. हम वापस स्लाइड शेयर कर रहे हैं जस्ट इन 1 मिनट. Hello.
Yes, sir.
Sir, you can proceed, sir. Sir, you can proceed, sir. Slide is there.
Slide is there. Actually,
Yes, sir.
I am starting from the beginnings.
Okay, no problem, sir. Slide is there.
Before the starting presenting my lecture, first of all, I would like to introduce myself. I born in a farmer family on March 1964 in village Bibipur, via Kandrapur, district Azamgarh, and my school education was completed in the village schools. After completing the school education, then I took an admission in BSc Agriculture program at Narendra University, Faizabad, where I was at 19 from the 1983 to 86. After that, then I shifted at Govind Ballabh Pant University, Pantnagar, for the post-graduation program, and where I was from the 1989 to 87 to 89. And after that, then I shifted Varanasi University for PhD program, where I was from 90 to 92. As per concern the experiences, I have more than 27-year experiences on education, research, and extension. I started my career from the assistant professor at Indira Gandhi Krishi Vidyalaya, Rajpur, where I served more than 7 years from the 1993 to 2000. In May 2000, I entered in ICR system and joined as a senior scientist at ICR Research Campus of Barapani, Meghalaya, where I served more than 5 years from May 2000 to August 2005. Again, after 2005, I transferred at Indian Institute of Vegetable Research, Varanasi, and the same chemistry. In 2007, I became a principal scientist of professor, and still I am continuing in this institute. As per concern the research area, I am basically I'm a vegetable breeder, and I started my breeding program from the PhD degree programs, where I did genetics of field and quality based tomato. Still, I am continuing the tomato crop for the breeding program. I completed most from yield, from biotic and abiotic research breeding program, both traditional and molecular breedings. Besides this, I have also worked on the how to enhance the vegetable production through the plant growth regulators. I have standardized the plant growth regulators in some vegetable crops. I also took an opportunity to work on the organic manure, particularly for the standardization of the organic manure at ICR sector of Barapani, Meghalaya in collaborative modes. Besides, I also brought the 8 externally funded projects and generated the more than 16 crore rupees for the from 2000 to till date. And also I completed all in-house projects, where 8 in-house projects from ICAR Varapani and from the IIVR Varanasi. As far as we concern the achievements, I developed 25 varieties of tomato, cowpea, Indian bean, and French bean. Besides, I also identified the 42 lines for the different vegetable crops for the resistant tolerant to biotic, antibiotic resistance, and quality traits. Besides this, I also generated the 15 technologies of the different aspect of productions in different agroclimatic conditions. And for concern the publication, we published 130 research papers and in 16 international and 29 national journals, which are from the India, published from India, Netherlands, Poland, USA, UK, Italy, Africa, Thailand, Germany, and I also wrote the 5 books and 47 book chapters and 57 articles. For the awards and recognitions, and yesterday I got a letter from the president of the NOS Fellow for the NOS Fellow. Thus I became a national NOS Fellow in 2021. Before that, I was also the Fellow of the Society of Horticulture Research, Fellow of the International Society of Knowledge Sciences, Fellow of the Horticulture Sciences, Fellow of Indian Society of Vegetable Science. Inside this, I am the lifetime achievement achievers from LTC Lifetime Achievement and Dr. Harbhajan Singh Awards, Memorial Awards. I am also recipient of the Research Paper Reviewer Awards. Then again, I received Dr. Harbhajan Singh Award in 2015, HITH Gold Medal Awards, Year of the Scientist Award, Distinguished Scientist Awards, and Young Scientist Associate Award and Craft Research Award. The topic which we have chosen, that is a traditional and molecular breeding approach to manage the abiotic stresses in tomato. Before starting the lectures, good morning to all of you, respected participants and dear friends, ladies and gentlemen. Before the starting my lecture, I am grateful to my beloved teacher, Dr. R.K. Pathak sir, by which I could understand what is horticulture in NDUAT Faizabad, today is called Ayodhya, from 1983 to 86 in my graduation degree program. I am also thankful to Dr. S.K. Dwivedi, who was my co-advisee at GBPUT Pantnagar, Uttarakhand, for giving me an opportunity to express my 30-year experiences on tomato. The topic which I am going to present is also a part of my research work, that is a traditional molecular breeding approaches to manage the abiotic stresses in tomato. I regard the tomato, which is considered as an ideal research vehicle or material because of it is easy to culture, as a short life cycle, high self-fertility and homogeneity, great reproductive potential, high amount of genetic diversity exists. In case of tobacco, more than 1,300 genes have been described, and out of which 242 have been assigned to a specific location among 12 tomato chromosomes. Normally, tomato is self-pollinated, but natural cross-pollination also occurs. In California, it is reported that the natural cross-pollination occurs 1.9 to 47.1%, whereas South Peru, it is reported that natural cross-pollination occurs 15 to 20%. Globally, tomato is being grown in 3.7 million hectares with production of 200 million with production of 200 million tons. Mexico, Brazil, Italy, and Spain are top producing countries in the world. India is the second largest producer in the world, which contribute 9% of major vegetable crops. In India, 13 states are major producing states, out of which Andhra Pradesh, which contributed 13.99%, whereas Madhya Pradesh contributed 12.24%, and Karnataka contributed 10.53%. Though there is tremendous research work has been done in this crop, but average productivity of tomato is very low. That is only the 15.9 ton per hectare if compared with the watt productivity, which is a 28 ton per hectare. Tomato productivity can be increased up to 80 tons per hectare. The USA has reached up to the 70 ton per hectare. As per concern the nutrition, tomato ranked 16th position as a source of vitamin A and 13 ranks as a source of vitamin C. Due to high nutritional profile, tomato is also called for poor man orange in India. Besides nutrition value, this crop has also medicinal value as it helps in control of prostate cancer, improve the vision, high blood pressure, good for diabetes person. For processing point of view, this crop is ideal crop as several processed products like ketchup, sauce, puri, juice, powder are prepared. During the 17 and 18, India exported tomato and earned rupees 11,421.9 lakhs among the vegetable crop. Solanaceous vegetables, including tomato, are a good employment generation, especially common, particularly for hybrid seed production, whose contribution is 56.46. Tomato suffered several biotic stresses like temperature, both high and low, moisture, deficit and excess, salinity and weeds. Here we will discuss only on the temperature, moisture and salinity. In tomato, heat tolerance is measured by the ability of a plant to step fruit at a high temperature. When temperature goes beyond the 32 degrees centigrade, depending upon the variety, fruit set is reduced. High temperature or fruit set is a general problem in tropical and subtropical part of the country. Tomato is sensitive to low temperature and generally most of the plant growth and development are adversely affected under cool temperature conditions. When temperature goes down to below 12 degrees centigrade, uh, there is a low food setting. The ability to germinate tomato at low temperature is controlled by a recessive gene is known as LPG and which is a polygenic recessive in nature. The percentage of food set at low temperature is controlled by additive, additive and dominance component. Therefore, recurrent selection for breeding is being used for development of variety of lines tolerant to low temperature. Isozyme markers associated with cold tolerance, that is a PGIL, which are located in chromosome number 6 and 12, has been reported in a wild species, Solanum haplotype. The essences are LA1772. Orthocarpic variety can be also grow in the low temperature because in orthocarpic variety they have very good potential to set the flowering and fruiting in a low temperature. And the gene which are responsible for the low temperature is that is known as PAT gene. Though tomato is sensitive to both chilling and freezing temperature, but it is not a problem of the northern part of the country. It is a major problem in the hilly area of the country. Healing is measured by germination of the seeds, plant growth, photosynthetic rate, fruit set, and quality. However, freezing is measured by what winter hardiness, winter survival, frost resistance is measured by hypocotyle growth rate. The marker loci TGI is only loci to have protect the effect of low temperature. After the temperature, then moisture stress. Generally, three terms are used for the moisture stress: drought tolerance, drought avoidance, and drought resistance. Terms are generally used. But tomato is facing only the moisture excess and moisture deficit. The avoidance term is referred to as a drought tolerance with the high tissue water potential. Drought resistance is the sum of drought avoidance and drought tolerance. Drought stress resistance is measured by attributes like germination percentage, rate of seedling growth, stomatal control of transpiration, leaf area, root growth, proline content, and yield. In oil species Solam pilli has been reported that very good germplasm या material which can use for the breeding program for the drought resistance because of their morphological characters. Generally, direct selection method is being used to identify the resistant assistant or lines. Under the excess moisture conditions, the production of tomato is very difficult because due to waterlogged condition, there is reduction of oxygen supply to the plant roots, which results an accumulation of the exogenous ethylene in the plant and causes the impedency. And due to the waterlogged situation, what happening? There is a anaerobic which develops, and due to this region, there is a displacement of oxygen from the soil and soil pore space. Under the waterlogged condition, there is a disbalance of the hormone between root and shoot, with root and shoot. In tomato, flooding drastically reduced the level of depredicacy in the xylem sac of the plant. Under the anaerobic condition, there is a formation of some toxic substances like sulfur dioxide, sulfur hydrogen sulfide, and hydrocarbon, which causes the toxic to plant. And due to this region, there is a dam amines building of the plant died due to the waterlogged condition. As concerns the genetic materials, still there is scarcity of the germplasm in terms of the variety, in the term of the wild species, which can survive in the waterlogged condition. Very early, one line has been reported, that is a Nagendra, and this line has been reported in Philippines, and this is the means derivatives of the Solanum cerassi army. Yes, tomato for the tomato production, if we see the uh, tomato production, each stage of the tomato is affected by temperature. And in case of tomato, night temperature is more important as compared to the day temperature. If temperature goes below the 10 to 12 degrees centigrade and beyond the 35 degrees centigrade, there is no flowering fruiting because they require the specific temperature for the flowering fruiting. Like if temperature is less than 10 degrees centigrade, fruit unable to develop the red color. If temperature is between the 10 to 20 degrees centigrade, optimum for good for color development. Temperature of the 21 to 24 degrees centigrade, it is best for the lycopene development. Temperature less than 18 degrees a day and 12 degrees in night causes the chilling injury, and due to which there is a low fruit setting. And for the good fruit setting, 21 degrees centigrade temperature is very optimum, and this situation comes in northern India in the month of the November. Therefore, for the hybrid seed production or the this is the best seed time, and this is the best month for the hybrid production. The temperature goes 25 degrees in day and 18 degrees in night, there is a best for the flowering and fruit setting. If temperature goes more than 30 degrees, the red color disappears and then yellow color comes. The more than 32 degrees centigrade day temperature or 22 degrees night temperature affect the fruit set adversely. If temperature goes 40 degrees centigrade, the lycopene means red color. Lycopene, due to the lycopene, there is a red color into where this appears. How now question is how the temperature affect in the plants, and it affect on the start from the seed germination. Due to high temperature, there is no seed germination. It also affects the seed growth and flowering, fruit set, ovulation, endosperm degeneration. A number of the consequences arise due to the high temperature like splitting, intercode, but drop. Here I like to explain that what happening due to the high temperature. Due to the high temperature, there is a stigma accretion, and due to the stigma accretion, what was the pollen release? They could not land on the stigma. Therefore, there is no pollination, and due to no pollination, there is no food system ultimately power drop. Now, in case of the low temperature, what happening in low temperature, there is a formation of endothelium layer in the anther sac, and due to this formation of endothelium layer, there is no release of the pollen grain from the anther sac, due to which the stigma could not get the pollen, and there is no pollination. Then ultimately there is a drop of the flowers. This is the situations happening in the high temperature and low temperature. Now, what are the mechanism for the high temperature? And this mechanism is there are two mechanisms are involved in the high. First is the heat avoidance, and second is heat tolerance. In case of heat avoidance, it depends upon the transpiration, and it also depend upon the leaf characteristics. If leaf is in pubescence and glycogens, then and this reduces the light absorption and also reduces the heat. And insulation of bark is also responsible for the heat avoidance. In heat tolerance, usually improves the membrane. Yes, heat tolerance. There are number of factors involved. That is a membrane stability, stability of photosystem second, photosynthesis translocation, system reserve mobilization, and osmoregulation, and all the things are associated with the heat shock protein and also causing the hardening. In heat tolerance, usually improve the membrane stability under the heat stress. Stress. Membrane stability is determined by two factors. First is the fluidity and lipid fluidity and electrolyte leakage. In general, cell membrane thermostability is a fair index of the genetic variation of heat tolerance. Heat tolerance. The effect of heat tolerance on photosynthesis and chloroplast damage can be assessed as variable fluorescence at 685 nanometer. Keloid formation in the phloem sheep tube is the major for the inhibition of the translocation by heat in tomato. There are some osmoregulators which are formed during the high temperature. These osmoregulators, like the proline and glycogen, betanin, they are also causing the heat tolerance, and they act as a protectives or in heat stress conditions. And if you are applying these osmoregulators, then the ability of the fruit setting and falling are increased. The, the association among the reported trait under the heat stress, the genetics of the stigma position are the important for the understanding of the genetics of the food set and high temperature, because there is a strong association between food set at high temperature and position of the stigma. The position of stigma is determined by a few genes which is influenced by the environment factors. There is a preponderance of additive gene action of root set, flower drop, and underdeveloped ovary, while the plant height and quality parameters like locule numbers, TSH, ST, and lycopene content dominance are more important. And in case of the tomato, what we have observed that most of the food characters are responsible for the heat stress. They have a preponderance of the non-additive gene exons, hence indicating that there is a means heterosis breeding would be rewarding uh, for the development of the lines or hybrids for the high temperature tolerance. If they see the heritability in the narrow cell, this is the low heritability. It means there is more influence of the environment factor, thus is indicating that there is a less scope of the possibility. There is a less scope of selection and more scope of the possibility. As per concern the hypothesis over the better, it has reported that for the food set, it is a 53.78%, where the pollen viability it is 92.58%, and food cost per plant that is 81.48%, and for the yield it is 116.6%. Before, when we are going to start the breeding program, we also should identify the what characters should be the donor parent for the abiotic stresses in tomato, and what breeding procedures the characteristics of the donor parent should be. Must be the short lifespan, which help the minimizing the temperature effect of palm. Awareness of the leaf will provide the positive state of the cell wall, cell membrane, repel the sun ray. Leaf orientation, small leaf size, long root length, and potato leaf type, cuticular wax, and leaf rolling. And it must have the moderate heritability, and that should be the positive association of field of stress. Back cross method of followed by recurrent method is breeding is being used for the development of the breeding lines tolerant or resistant to the abiotic stresses in tomato. And this is the schematic presentation how we proceed and how to start the transfer of resistant gene in any susceptible variety, and how much we are getting the means genes are coming in the generation to generation. Now, screening, what are traits which we are using for the screening, and there are some phenological characters, that is a pollen viability, stigma insertion, empathy burning, percent food checking, number of foods per plant, yield per plant, and in physiological parameter, relative water content, electrolyte leakage, chlorophyll fluorescence, and total moisture. In biochemical, what we are using: chlorophyll, phenol, proline, lipid peroxidation, hydrogen peroxide, catalase, superoxide dismutase, ascorbate oxidase, and glucinate reductase. And the molecular we are using for the fruit setting, we are using the scale of the 1 to 5 scale. If the fruit setting is 0 to 20, it comes in highly sensitive. Likewise, in fruit setting more than 35%, it comes the highly tolerant. Genesis for the identification of heat tolerance, how we have started the uh, high temperature tolerance research on tomato. It was started after the launching of the NICRA project at IIVR Varanasi. For screening, what we have done, first we have collected the 820 jar plant for the different organizations, and these lines were screened in the field twice, and then we have selected 250 genotypes from the 820, and again then these 250 genotypes were tested, and we have screened the 30, and 30 among 30, then we have screened the 9 genotypes which we have found the tolerant, means high temperature, means more than 35 degrees centigrade degrees centigrade. And these lines were tested in TGT artificial screening, then we found only the 4 line have ability to set the flowering protein in low temperature, in very high temperature, that is more than 38 to more than 38, and in between the 30 45 degrees. And we are using these our lines as a donor parent for the development of the hybrid and variety. Yes, what sources are available for the item heat tolerance? We are trying, if we are talking about the wild species, sorghum is the best source for the for developing heat tolerance. In past, it means during the 90 era, number of the lines or varieties which have been identified by the Dr. Kallu at Haryana Agriculture University and this group, and they have reported that HS1012, HS101, Margino, Red Rock, Walter, Switzerland, several in Punjab, topic and selective. Several in the and selective is a very best good material, and they have a facultative party. They can the material can also be used in both heat tolerance and low temperature. At IIVR, we have also identified some lines or some varieties which have ability to set the flowering protein more than 35 days. These are Sancheri, EC 538880, Superba, CLN 16221, and CLN 2026, and these both material CLN we have received from the material from ABRTC, PR and 168, and these material we have generated from the real populations. And H88 and 78, this material was also very good material for the developing the variety for heat tolerance. How to we have proceed for the develop the breeding program in the first we have developed the hybrids, and first we have developed the 456 hybrids into line replacement material, and these 456 hybrid we are evaluated in both conditions, in field condition and TGT. Finally we found that VRNT at 18283 VRNT, these are hybrids which have relative constant giving the heat tolerance, and they and giving the marketable foods up to the last picking. This is the hybrids which we have got up from the 456 cross combinations. Here we have taken the three criteria. First, our flowering fruiting must be in the month of the April and May. Second, the yield should be more than 35 tons, and the size of the fruit should also maintain up to the 3 to 4 feet. Because in case of the high temperature conditions, the size is very most of the hybrids or most of variety, they are giving the good fruit size in the first picking, but after that the size is reducing. Therefore, this criteria we have taken, and on that basis, we have found that only one hour hybrid, that is VRNT at 18283, they have giving a more yield, that is 35, more than 35 tons per hectare, and the fruit size is also not much more reduced up to the last picking. Last picking means up to the 4 to 5 pickings. We have also compared these hybrids from our private sector bred hybrids, which are generally grown in our Varanasi conditions. Though we have taken the 20 hybrids, but here we are presenting only the 7 hybrids which are performing up to some extent good in high temperature conditions. And here our hybrid we have offered where the foods are available up to the June third week, and here the private sector hybrids, they are only fruits are available up to third week of the May. One thing I'd like to say that when we are talking about the summer tomato cultivation means transplanting in the first week of the February and putting fruits availability up to the by June, because there is big confusion. Some people are coming and sir, we have also a high temperature tolerance variety and hybrids. And when I asked the question, when we are transplanting, so they are saying that we are transplanting in the month of January and we are getting the fruits up to the appointment. Now, whatever I am understanding, they are flowering or fruiting are occurring in the March, and fruits I already set in March. In the March is very congenial temperature for the fruit, flowering fruit. This will be not in the hybrid variety for the high temperature tolerance. This is our hybrid which we have in the field conditions, and this is the color development in our hybrids. And this is the picking wise how the first picking what we are getting, and second picking what we are getting in whatever hybrid we have tested. One rest, this is the how the reduction of the food weight in the hybrids food weight in our hybrid, which we see the hybrid which we are TH 203, we are getting the constant food weight up to the fourth picking, but after the five picking, their side. That's why we are selecting this hybrid. And we have also we tested for the shelf life, and we have found that this hybrid is giving very good shelf life with the minimum physiological weight loss. Is the weight loss of the different hybrid. Now, this is the color development. See the color development, how the color is developing. Here, the color development up to we are getting the very red color up to 14 days of the storage, and other hybrids you can see even the 12 days we are not getting very good color. This is the we have given the this is our hybrid which we are going to recommend, we are going to apply for the summer cultivation, that we have given the name, and that is Kasia Dut, and it is also registered in NVPGR, and this hybrid is giving the more yield, 54.5% more yield as a private sector bed, that is RL60, because this is the only hybrid among the hybrids which is giving the more yield in summer season. That's why we are using as a check hybrid for the testing program. This is how we can test purity testing. We have used the more than 45 SCOT markers, and these SCOT markers can be used for the testing of purity of the Asiadut VRL PH 18283. This is the how for the this data for the knowing the expression profiling of the gene, responsive gene. We have used the two lines, that is H88S78S1 and CLN1621 as a resistant material, where Punjab Chwara as a susceptible material. What we have observed that most of the phenotypic stress tolerance, both genotype and biochemical basis, on the basis of the biochemical, what we have observed that the water content, photosynthetic pigment, free cellular protein, and antioxidant molecules, they were very higher in the resistant variety like H8870-1 and CLN1621, whereas it was very low in Punjab Chwara. As for concern the hydrogen peroxide and other electrolyte leakage, it was very low in the resistant lines, and whereas it was higher in the susceptible line. In heat shock protein study, what we have observed that there were 67 genes were responsible for the heat tolerance in genotype H8878-1 and CL1621. In QTL mapping, what we have observed that each 67 the genes which are present in our line H8878-1 and CL1621, they are equally, they are distributed on all the 12 chromosomes, and all the stress responsive genes, they are present in cluster in terminal. Most of they are present in the terminal portion of the chromosome. The number of the chromosome vary from it was the minimum in chromosome number 5. We see the chromosome there are minimum, whereas the maximum on the chromosome number 8, it was 10 means genes which are available on the chromosome number 10. As for concern the moisture deficit, tomato can be grown in rain-fed condition even without irrigation from the November to first week of February. A tomato can be successfully grown in Varanasi conditions, but present day there is a wide water crisis is going on, particularly in the states like Maharashtra, Dadar, Karnataka, Andhra Pradesh, Odisha, Gujarat, Telangana, and Rajasthan. In case of tomato, three stages are very important for the moisture point of view, that is flowering stage, fruiting stage, and fruit development. If there is a crisis of water, then it will affect on the yield. And due to the water stress, what happens? There is a power setting, lack of fertilizer, reduced food set, and fruit spreading, puffiness, poor seed ability, and blossoming. Now, what scale we are using? We are using the 1 to 5 scale for testing the our lines or varieties, and if under scale it is highly proud. And this is the what symptoms are coming in the plants. On that basis, we have categorized and drought tolerance, average drought tolerance, and highly drought susceptible. And these are the correct symptoms in the plant. On that basis, we can categorize that this is the drought tolerance, this is the drought tolerant resistance. This is the mechanism. I have already explained the mechanism, what are the various mechanisms involved for the drought tolerance, yeah, moisture condition. Now, how we are proceed for the breeding program for the moisture deficit? If we are talking about the wild species, two wild species are very important which we can use for the breeding for the moisture for development of lines or varieties for the moisture that is Solanum many and Solanum 50. Earlier, some lines which have also reported by Haryana in Haryana, Dr. Rana and Kallu in 1990, that these lines can be used for as a breeding materials, whereas the IHR has also reported Arka Meghali, and now it is being used as a breeding material for the developing varieties and lines for the rainfed condition. At IIVR, we have also screened the material by using the suitable procedures and for the screen, and we have still found that each material has ability to give the very good yields up to the 50 to 60 days without irrigation. And these material we are using by the breeding material, and on that by use of these materials we have developed this 40 hybrids, and we have tested in the moisture deficit condition, given the moisture deficit condition. We have also tested the both in the moisture deficit condition and irrigated condition, and compare the how much the yield is reducing in moisture deficit condition. So we found that the VRTH3, which reduction is very low, that is a 25.94%, followed by VRTH5, that was 28.35%, whereas in VRTH it was 29. Other hybrids which we have found that more than 60% reduction in yield, and then we have discarded those hybrids which are showing more than 50% reduction in yield, and we are only keeping those hybrids which have reduction in only the less than 30. So on that basis, we have selected only 3 hybrids. That is, this is our hybrid which we are planning to popularize among the power, and you can see how the food setting in this hybrids, and we have not given the 60 days up to 60 days of the water in these plants, and because we have transplanted in the month of October up to the vegetative growth, we have given the irrigation, and in after the November, first week of November, we have not given a single drop of water in these hybrids, and even though they are giving a very good yield and the yield potential of the 59 tons per hectare, but when they are grown in the irrigated condition, their yield potential is 143.33. Only the reduction actually is 37.9%. So thus we are recommending these hybrids for the rain-fed condition, or those areas where the moisture there is a crisis of moisture. Now we have this by we are trying to prove why this our hybrids are showing the resistant or tolerant to the moisture condition, and on the basis of the physiological parameter, particularly membrane integrity. And if we found that the both hybrid VRTSC that relative water content were in decrease the electrolyte leakage. Same things, moisture deficit, photosynthesis, we have also recorded the photosynthesis in terms of the chlorophyll and peroxide. We have found that the VRTS that they have the means increase the photosynthesis and chlorophyll peroxide as compared to the others. Same thing, we have also recorded the proline content, reactive oxygen species. That we found that VRTS that has a more maximum increment as compared to the other hybrids, term of the proline and enzyme catalase, and they are both are increased in these hybrids. Effect of the moisture deficit nutritional quality, yes, we have also recorded whether the stress is affecting the nutritional quality or not. Then we found that the nutritional quality is increased in VRTS, that the maximum in the hybrids as compared to the hybrid. Next, actually, this data we have recorded from the Bioinformatics Center from Varanasi University. Just we are trying to prove that which gene, what are the genes responsible for the stress in moisture stress in these our hybrids. So we found that NAC1, NAC1.1 and 2, they are responsible for HSP and LA and EFP, they are responsible for the moisture deficit, and we found that their contribution is maximum contribution in VRTS16, that's 3 followed by 68, and NAC means non-apical meristemic versus transcription activation, HSP means it's a protein, and LA means a late endogenous dependent protein, GFP means in fingerprinting protein, and EFP elongation finger protein. And actually, this data we have received from the Varanasi University for the bioinformatics centers, and thus we have proved that our material, whatever we are material are focusing, they have a ability to the moisture deficiency. Again, we have also analyzed the phylogenetic testing, and we found that since prolonged as we reported that this is a tolerant to can be used as a source material for the developing the tolerant to moisture deficit condition, and we have compared that this is matching of the solar tuberous. Again, this is they have taken the gene structure and conserved motifs under the moisture deficit. We found that there are four motifs which we have identified, and the strong conservation around the VKT domain across all the basic protein. DNA protein backing we have also by this we have also tried to prove our material that they are superior, or there is involvement of the some protein that is arginine, amino acid, proline, arginine, trypsin, lysine. These are the responsive amino acid for the stress of the molecule. Yes, saline tomato cannot be grown in saline soil, and it requires the optimum pH of is 7 to 7.5. It can be grown, but if the temperature goes more than 7.5, then it is very difficult to grow the tomato. And I think in the India, all states which are facing a lot of problem of the salinity, particularly Andhra Pradesh, or Gujarat, Haryana, Karnataka. And in case of the salinity, it can be classified in several types, like slightly, and it depends upon the exchange of cations and pH level and sodium percentage. And this is for genesis how we have started the work on the salt tolerance. Here we have first started from the seed germination, and for that we have taken the 120 genotypes, and then we have seen in the four concentration of the salt, that is NaCl and 40 micromole, 60 micromole, and 2 and then we have see the germination. After the germination, testing the germination, and in 120 we found that the FLA 717, Mount Probate selection 7, KT18, Angur Lata, and Agita 30, and these lines we are found have ability to germinate in the very high concentration, that is 100 micromoles of the NaC. Then we got a clue for the tolerant or resistant, tolerant to salt lines, and we also tested these lines in the pot culture, and this is the symptom of the salt affected lines, and this is the salt tolerant symptoms. And we are using the scale and 1 and to 5, and these are the characteristics of the plant, and on that basis we have categorized the materials, and finally we have again go for the hybrid development, and we have taken the what were the materials we have tested, and as a male parent and female we are using, thus we have developed the 32 F1 hybrids, and by using the donor parent, and these hybrids were evaluated in two phases. First phase we have evaluated in both concentration from 200 micromoles and 400 micromoles, 200 micromoles, pH 7.5, and 400 micromoles means pH is 8.3. And both we have tested, and we got that number of the hybrids were able to tolerate the 200 micromole of NaCl. After that, for squeezing of the materials, then we again tested in the 400 micromole initial, and whatever we observed that those materials which have capability to tolerate the salt up to the 400 micromole, they have they have, we have kept in our considerations, and these hybrids which we have observed that they are they have capability to tolerate the salt up to 400 micromole, and pH is converted in pH, it comes in the 8.3. It means whatever materials we have only the tolerate the 8.3 pH. This is the performance of the whatever we have selected hybrids in under the salt condition. And we have also third thing we have tried why these our hybrids are very tolerant to this is the reason right to the physiological parameter that is membrane stability. We have observed that maximum damage of membrane stability in terms of electrolyte leakage that was 5.2 15% and lipid parameter 42.1 and 55.6% was observed in hybrids that is VRTS 17.47 and 17.49. However, the hybrids 1754 and 1755 it elevated the salt-induced membrane damage, showing the minimum electrolyte leakage that was 2.2 to 7.8%, and lipid parameter was 36.6%. Same thing we have also tested relative water content and photosensitive pigment. What we have observed that we are TH 17-47 and we are 17-55, so the better holding capacity as they give the minimum decrease that is a 2.1 to 3.1% relative water content in the salt, whereas the chlorophyll content and peroxide content we found that only 62.2% and 29.5% in all hybrids under the salt. The hybrid VRTS 17-54 and hybrid VRTS 75 so higher chlorophyll and carotenoid content as compared to the other hybrid under testing. This is also we have taken the proline content, and this is the catalase activity. What we have observed that proline content decreased positively by the 49.4 to 130.6%, where the H2O increase in generation increased significantly 39.1 to 60.1%. The hybrid VRT 17-55 and VRT 74 showed increased proline and hydrogen peroxide content under the salt test compared to the other hybrids or other control materials. This is the here we are also tested how much yield is reducing if our materials are grown in without salt and in salt. So we found that our two material that is VRTS 17-49, their reduction of yield is 46.2%, whereas the VRTS 17-44 a reduction of the 55.6%. Then again, we have received this data from the Bioinformatics Center on our Singh University, and what are the genes are responsible for the salt tolerance in tomato. Particularly, these are the genes which are responsible, and how much contribution occurs in our what were the tolerant material that is in F gene that is 75% and 39%, but F2 24 79%, F3 74.4%, where ATP that is 82.4%, and ZEP FP that is a 71.4%. Likewise, EFP that is a 42.3% and 62.6%, depending upon the. Same thing we have also found that our material when we have and see these phylogeny, we found that this material are matching with the solvent is close related with the solvent progression. Same thing like the drought tolerance. Here we observed that 19 conserved motifs were identified, which showed the strong conservation of AP2 domain across all the number of driven proteins. This is same things which we are, and these are the amino acids which are responsible for the salt tolerance, particularly or whatever material we have generated. Next is the as per concern the QTL mapping for the QTL mapping, what we have observed that 76 QTLs we have identified, and these QTLs are evenly mapped on chromosome number 12 chromosome number and showing the uniform distribution except the 7 and 8 chromosome. 7 and 8 chromosome, where most of the QTLs are associated with the middle part of the chromosome, and among the entire QTL, maximum QTL was recorded in the chromosome number 1, where the 17 QTLs are found, and followed by chromosome number 6, where we observed that there were 13 QTLs, and in chromosome number 3, there 12 chromosomes are associated for the salt tolerance in tomato. This is the all the pattern of the means gene responsive gene available. Yes, tomatoes cannot be grown in a waterlogged condition. If there is a few hours water stagnation in the field, the plants die immediately. And we see the rain pattern during the 2011 and 2012 and 2019. There is a accidental rain. It was not, we were not hoping that in the month of last week, last week of September, there will be such a high rainfall. In such situation, it is very difficult to grow the tomato because at that time tomato crop is either in the vegetative phase or the transplanting phase or northern part of the country, especially in Varanasi. What we have observed for that, you can see the position when in such situation, all the area is somewhat, and it is very difficult to grow the tomato or to manage this situation. What we are thinking, we are thinking to promote the or evaluate to promote the rainy season varieties or hybrids. Or second thing, grafting is also one of the way by which we can manage this abiotic stress. For rainy season, what we have observed that we have evaluated 250 lines, and these two lines, these two temperatures are evaluated for the 3 years consequently in the fields, and and after the 3 year, the 3 year of the evaluation, we found that these are lines BRT 34, TLC this, BRT 50, BRT 51, BRT 01, BRT 30, and TLC 16-1, and selection 7, and RC Aman, Punjab, Barkhawan. They have success, they can accept the selection 7, where the tomato leaf virus is a serious problem during the month of the rainy season, and other materials they are performing very well for the rainy season. In case of the summer rainy season, what we have observed, farmers are facing uh, 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 both biotic and abiotic stresses. In biotic stresses, they are facing the fusarium build, bacterial blight, tomato leaf curve virus, and fruit borer. And in abiotic stress, in up to say August, September, the temperature is very high, and in such situation, there is no food stream. Therefore, we have taken the consideration of what are the material, their ability to set the uh, uh, fruit setting in the month of September. Then we have taken these material our consideration. Second point that we have also taken in consideration that it must be the resistant or tolerant to the tomato leaf virus. And one thing, how much reduction in yield if we are cultivating in the Rabi season. So what we have observed that this is the one material which a reduction in wind yield, and if we compare with the yield in the Rabi season, and this is the if you are grown in August season, the BRT 51 and 52 reduction is very, that is only 2.1%, whereas BRT 50 and 38.3%, and in 32 this is also 1.7%. And one thing, these materials, BRT 50, BRT 51, BRT 30, TLC60, they are possessing the TY3 gene. They are resistant to tomato leaf curl virus because in case of the northern part of the country, you see that this gene is responsible for the resistant to tomato leaf curl virus. And we have also, this is the other more infiltrate culture, and we have also evaluated the hybrids, and we have taken 91 and 22 hybrids from the IRI, and what we have observed that these hybrids, they are performing very well in the rainy seasons, and we can grow successfully in rainy season cultivation. Yes, grafting is also a very new option for the to manage the abiotic stress like waterlogging condition. And this technology is not new technology, and it was first used in Japan and Korea in 1920, and in India, this grafting technology is being grafting technology is being systematically in research work initiated at Tamil Nadu, Coimbatore, and Varanasi, IIVR Varanasi, and IHR Bangalore, and Jodhpur. What we are doing here, and we are all standardizing the root stock. We are also standardizing the sign, and we sign for the manage the waterlogging condition and moisture, waterlogging condition and moisture stress in tomato. So under the root stock, we are using the IC 354557, IC 111056 Surya, Solar Microcosm, Ethopicum, we are using our the inside variety, Kasi Yamun, Kasi Ada, and Kasi Chen, we are using, and this all the products are going on under the ICAR project. In my institute, Dr. Anand Bahadur, my colleagues, is working on the graphic technology in our institutes, and this is the just a for the point of view. Just we have given what are the root stock and sign variety are available in vegetable crop, including the tomato. In tomato, we are using IC, I've already told, IC 111056 and IC 3545%. But and sign variety we are using for the also using, and IHR people have also used these materials, and this is a various root stock including tomato, brinjal, and here the hot pepper, and here the sponge guard and bottle guard for the both the biotic and some abiotic stresses like waterlogging, flooding, salt, and drought tolerance, and also the for the cadmium and nickels and some of the micronutrients. And these root stock and sign can be used in grafting technology and solve the problem of the abiotic stress in tomato and other vegetable crops. What we are root stock, we are using IIVR policy, and we are doing a very extensive work in tomato and other crops. In tomato, I will told we are using these are the root stock, and our specific objective to tolerate the waterlogging condition found in tomato. And we are beside the tomato, we are also working on the bottle gourd, watermelon, and brinjal, and other rich gourd, ash gourd, and we are using for these are the our specific are the use of the root stock. Now, what are the effect of the root stock in the sign variety? Yes, it has observed that every root stock is not applicable for the each sign. It differs sign to sign. Performance of the sign variety also differed with the root stock. Next is how the quality is affected in using the captain technology. There is a good way that root stock is affected in the quality case. So in the fruit crop there is number of literature of available regarding the effect of the root stock on the quality. But in case of the vegetable crop, particularly tomato, what we have observed that quality is not much more affected if we are using the brinjal root stock and using the sign tomato is sign variety. Yes, there is little bit differences in the size of the fruit, if we are using the brinjal stock having the oblong shape, and if soil variety of the flat so little bit there is a means shape it change. We are getting the fruit of the oblong shape of tomato. Yes, this is the performance of the grafting, how the grafting combinations and their yield potential in the grafting combination. Yes. When I was in graduation program, just I was listening that tomato. But today I am seeing what is the tomato, and if we just our scientist Dr. Anand has done very commendable work at IIVR Varanasi. At one same plant we can get the both potato and tomato. And same thing we have also done in tomato and brinjal. A new achievement of our institutes, and this technology we are using for the means both solving the problem of biotic and abiotic. This is the our means how we are till date. We have distributed more than 10,000 tractor farms to the vicinity of the Varanasi for the farmers, and we are promoting the electric technology for the solving the abiotic system like water line tomato. This is the yes, though we have everything are available, but we see the achievements regarding the abiotic stresses in tomato or any vegetable crops. The informations are very less. Reason behind this different concept regarding the abiotic stresses, inadequate genetic resources. There is no simple score reliable to dependable selection criteria for abiotic stresses. Negative correlation of yield with the abiotic stress, complex and polycyclic, poor heritability, and resistance and tolerance is poorly understood because genetics we have not much more studied in abiotic stresses in vegetable crops as a whole and tomato as a particular. Now, what should be the future strategy? For the future strategy, we should apply the wild taxa as much as search of the novel gene for a particular abiotic stresses, synergy of the physiological, molecular, genomics, and bioinformatics, establishment of the balance between these stress tolerance and high yield, enhance physiological efficiency for the chronic yield, exploitation of escape mechanism, and use of biotechnology tools. And this can be done by a very good team, should be a good team that is physiologist, chemist, molecular biologist, plant breeder, and bioinformatics, or the generating the basic strategy applied antiseptic research in abiotic stresses in vegetable crops as a whole and tomato as a particular. And these are the publications, though we have published more than 20 such papers, but here I am only reflecting those papers which have as a NOS rating of more than 7.10. And this is just our the some since this program is also attending by the some our students, they can read each publication and take the advantage of the our research and start the work accordingly on the to solve the abiotic process in vegetable crops as a whole, tomatoes in particular. This is our aim by which we can put that in, we could do the research and in abiotic stress and tomato, and these are the very good tips depending upon the our the DICRA project and CRP projects, and these are the our research fellows which have contributed a lot of informations, particularly generation of the science in abiotic stress in tomato. And I am thankful to these our young scientists. I can say the RSR, they are the most of us have become a scientist in the different organizations, and also the thankful to the our head of divisions and directors for given a very good opportunity to do the, to work in our institute. Thank you to all of you for listening patiently. I am thankful to again the organizer of the progressive horticulture, particularly the president, Dr. R.K. Pathak and Dr. S.K. Dwivedi, who have given me opportunity to share the what I did for the solving the abiotic stresses in tomato. Thank you to all of you. Thank you. Jai Hind, Jai Bharat. in the slideshow. हो गया क्या? ठीक है.
Hello, sir. Nagendra, sir. Hello, sir. Hello. Thank you for wonderful talk. Audience, please send your query and question to progressivehorticulture@gmail.com. Response of audience was tremendous with very encouraging remark. We are highly grateful, sir, to you for sparing time and delivering such an important talk. Thank you, audience, for your active participation and support. Thank you all.